Welcome. Today we explore the fascinating world of red blood cells, also known as erythrocytes. These are the most abundant cells in your blood, and they have one remarkable job: keeping every tissue in your body supplied with oxygen. Red blood cells are biconcave discs, like a donut without a hole. Their diameter is about 7 to 8 micrometers, and they are only 2 micrometers thick at the rim. Their unique shape gives them a large surface area, which is perfect for gas exchange. Importantly, mature RBCs have no nucleus and no organelles, so they have more room to carry oxygen-carrying protein. The red color comes from hemoglobin, which makes up about 33% of the cell's weight. Each hemoglobin molecule contains a pigment called heme, built around an iron atom, linked to a protein called globin. One hemoglobin molecule can bind 4 oxygen molecules. It picks up oxygen in the lungs, becomes oxyhemoglobin, and releases it in the tissues. The main function of RBCs is gas transport. They carry oxygen from the lungs to the tissues as oxyhemoglobin, accounting for about 97% of the oxygen transported in blood. They also carry carbon dioxide back from the tissues to the lungs as carboxyhemoglobin, roughly 30% of the CO2 transport. Additionally, hemoglobin acts as a buffer, helping maintain the body's acid-base balance. RBCs also carry blood group antigens and help maintain blood viscosity. Erythropoiesis is the process that makes new red blood cells. It mainly occurs in the red bone marrow. It begins with pluripotent stem cells becoming myeloid stem cells. These form CFU-E cells, which turn into proerythroblasts. After several divisions, the cells become reticulocytes and eject their nucleus. Within 1 to 2 days in the bloodstream, they mature into fully functional erythrocytes. Erythropoiesis is tightly controlled by a hormone called erythropoietin, or EPO, secreted mainly by the kidneys and also by the liver. When tissues are low on oxygen, the kidney detects this hypoxia and releases more EPO. EPO speeds up the maturation of proerythroblasts into reticulocytes. More RBCs enter the blood, oxygen delivery improves, and once tissues are well supplied, EPO secretion drops back. This is a classic negative feedback loop. Several nutrients are essential for healthy RBC production. Proteins supply amino acids to build globin. Iron is crucial for the heme part. Vitamins B12 and folic acid are needed for DNA synthesis and maturation. Their deficiency causes large, immature RBCs called megaloblasts. Vitamin C and copper help absorb and use iron. Bile salts help absorb copper and nickel, which also aid hemoglobin synthesis. Each red blood cell circulates for about 120 days. After that, its membrane becomes fragile, and macrophages in the spleen and liver destroy it. Hemoglobin is broken into globin and heme. Globin is digested into amino acids that are reused. Heme is split into iron and a non-heme part. Iron binds transferrin, is stored as ferritin, and returns to the bone marrow to make new hemoglobin. The non-heme part is converted to biliverdin, then bilirubin, then urobilinogen. Most is excreted in feces as stercobilin, and some is excreted in urine as urobilin, giving urine its yellow color. Doctors use several indices to assess RBC health. Hematocrit is the percentage of blood volume occupied by RBCs, normally 40 to 54% in males, and 38 to 46% in females. Mean corpuscular volume, or MCV, measures average RBC size, normally 76 to 100 femtoliters. Mean corpuscular hemoglobin, MCH, is the average amount of hemoglobin per RBC, about 27 to 33 picograms. And MCHC measures hemoglobin concentration inside the cells, normally 330 to 370 grams per liter. These values help diagnose conditions like anemia. From their biconcave shape to their oxygen-carrying hemoglobin, from their birth in the bone marrow to their recycling in the spleen and liver, red blood cells are tiny but mighty workers. Understanding them helps us understand life itself. Thank you for watching.